This is the second part of the video overview I'd like to give on biochemistry. For biology students and anatomy students who are just, trying to get a handle on all of the biomolecules which keep getting mentioned in later chapters. So kind of as mentioned in the first part, these biomolecules, they're big, and thus the fear is that they're complicated. But luckily, the way that many of these are made to such incredible sizes is that they have these little building blocks which just keep getting added one to the next to the next to the next. And so thus this huge molecule is just a chain of all of these smaller molecules, and thus more understandable. And so the building block which makes up proteins, and that's the first video went through carbohydrates and lipids. This video will go through proteins and nucleic acids as the four groups of biomolecules. The building block, the monomer which makes up proteins, these are known as amino acids. They're called amino acids because they have a central carbon, a hydrogen, an R group, which can vary. In the simplest amino acid, glycine depicted here, it's a hydrogen, but this can be a number of different molecules. And then this group called an amino group, and that group called an acid group. That's why they're called amino acids. And as mentioned in the previous video, that there's a type of reaction known as a dehydration reaction, where a hydrogen could be removed from one reacting molecule. An OH, a hydroxyl group, can be removed from the other reacting molecule. It's called a dehydration molecule because thus you have removed a molecule of water. But then these atoms then are now missing the atoms with whom they were sharing electrons. So they want to form new partners. And as they form a new partner and share new electrons, they form a new bond. In this example, this is what's called a peptide bond between the nitrogen of an amino group and the carbon of an acid group. And thus we have taken two amino acids and put them together to make what's called a peptide bond and forming a dipeptide. If we put enough of these amino acids in the correct order, this would then make a protein. Now, just as we can add amino acids together through dehydration reactions, we can split them in what are known as hydrolysis reactions. And in a hydrolysis reaction, that water molecule is put back, an OH on one reacting molecule, a hydrogen on the other. And so thus we can both build these molecules up through dehydration reactions, and we can and then we can break them down. Now, amino acids, there are 20 of them, and knowing which ones are reacting are important because there are differences between them. That's perhaps, a little bit beyond, the level which we need to look at these structures. But as, you start, thinking of, later topics, which amino acid is in which spot then makes a huge difference because the shape of the resulting protein is essential, and each of these amino acids has a different shape and then can allow for different shapes in the reacting protein. And so thus here you see that there are these 20 amino acids, and which of the amino acids we use matters. Some are acidic, some are basic, some are polar, some are non-polar. And so if you were, say, building a cell membrane protein, well, maybe the polar amino acids would be needed there. But if you were then looking at the part of the protein that's in the middle of the membrane, you would want non-polar amino acids. And so a protein is a chain of these amino acids, but which members of the chain you choose then becomes very important. Now, this video I'm going to kind of go through more slowly at the very end because it involves DNA as well. But very often, it's essential that at one specific part of a protein that there is, say, the amino acid proline and only the amino acid proline. Any other amino acid would then be potentially dangerous. And so when you ask, why does this person have cancer? Or why does this person have this genetic disease? It's because in a specific part of the protein, they're putting the wrong amino acid. So once again, some amino acids, they're polar. They interact well with water. Some are non-polar. Some are acidic. Some are basic. And if one were to replace by mistake through a mutation one amino acid for another, well, that could have then serious health consequences. And so if you're going to build proteins, they are built from amino acids. Amino acids are the monomers which compose proteins. And it very much matters which of the 20 amino acids you put together in that chain. And once again, the way that you join amino acids is by creating what's called a peptide bond between the amino group of one amino acid and the acid group of a second amino acid. And as we will see in the next chapter, this is occurring in the ribosome, a part of the cell where proteins are made. So in the ribosome, you bring this amino acid together with that amino acid, and you form a peptide bond as an OH is taken away from one and H from the other, and now they stick together. Now, this is such an important topic that it actually then just keeps coming up all throughout. So I'm trying to, go through every aspect of it. No, that would be inappropriate. So let's say you were building a protein to pump chloride across a cell membrane. So there's a specific chloride channel. You would want to put specific amino acids in a specific order. So glycine, then aspartic acid, then aspartic acid, and et cetera. So then this is representing the chain of amino acids that one would use. But you'll notice at one spot, if you make a mistake here, then this channel might not work. And then you could have, well, cystic fibrosis, which is one of the more common genetic diseases in the United States, which could actually end someone's life early. And so proteins are made of this chain of amino acids, but which amino acids you put in which order, that's important. So that spot right there in more than 2/3 of cystic fibrosis patients, that is the part that is where there is an error. In this next protein, this represents the P53 protein, which more than any other protein in the genome protects us from cancer. And so this is the correct amino acid. But every piece of that is where something is different than one. And as well as proteins do so many things. So for example, I mentioned blood typing in the previous video. While sugar groups, they are determining blood type, they're attached to a protein. So it's actually a glycoprotein. Other proteins on the cell membranes serve as receptors and antibodies, some of which recognize microbes and then can even be secreted to protect our bodies from microbes. So antibodies are proteins, many of which are on cell membranes, which allow certain cells, like our white blood cells, to recognize microbes and to respond to them accurately. There are other proteins of the cell membrane, which we'll see in the next chapter. Some are channels, which let molecules pass freely. Others are pumps, which can let molecules pass under certain conditions, etc. And so transport across cell membranes is very important. And proteins are one of the main ways in which that happens. Some cell membrane proteins are enzymes, such as those of the small intestine digesting disaccharides, say maltase and sucrase. There are proteins which compose the cilia and flagella, which are located on the surface of the cell, and they can be used in for movement, whether it be the movement of say, flagella, which moves the sperm cell, or whether it be, say, cells lining the oviduct or lining the throat, where the cilia are moving the mucus around the cells. Some proteins function as enzymes because in our bodies, we want chemical reactions to occur. So we want to take maltase in our diet and break it into the two pieces, which can be absorbed into the small intestine. We would like to take sucrose in our diet and lactose in our diet and break them into their component parts. And it's these enzymes, maltase, sucrase, and lactase, which break up those disaccharides. Chemical reactions can happen without enzymes, but they just don't happen quickly enough in most cases for us to maintain life. So here you can see that if we want to get energy out of sugar, we have to perform a large number of chemical reactions, and each of the green molecules coming in is an enzyme, the protein which is inducing this chemical reaction to occur. So what do proteins do? Well, a lot of things. So in addition to serving as cell membrane proteins on the cell surface, such as flagella, many are the enzymes which are causing chemical reactions to occur. So when our bodies, we make things, we break things down, we convert some molecules into others, all of these then require protein. We transport things in our body, and many proteins are then functioning in transport. So for example, hemoglobin in red blood cells is transporting oxygen and carbon dioxide. Other things in our blood they don't dissolve in water, and testosterone. Cholesterol in our blood, cholesterol carrying the three colors, the red, the green, and the blue, represent proteins which are in those lipid particles. As we can see in the next video as well, proteins can represent signals which tell cells what to do, in addition to receptors which receive those signals. And so we can make the signal erythropoietin, a protein, which then binds to the erythropoietin receptor, which tells a cell in the bone marrow, the cell, or cytokine hormones, which tell these bone marrow to go into various kinds of complex. This is cells cell and deciding who they when they grow up. And these signals which are sent are very important. And so proteins perform so many of the functions inside cells. And in future chapters, when we talk about the muscular system, then we talk about muscle proteins. When we talk about hormones, then we'll bring up the proteins which are hormones or receptor for hormones, etc. The last, and I'll get back to proteins in just a little bit as well after we're talking about DNA. The last of the four categories of organic molecules are the nucleic acids. Now, even though nucleic acids can involve big complex molecules like DNA and RNA, I'd like to start with a simpler molecule, which is a nucleotide with extra phosphates attached. So nucleotides are the monomers which will make up the long chains of DNA and RNA, the polymers. But here we see a very specific one. Here you see one nucleotide represented in this animation with two phosphates. And if we wanted to attach a third phosphate, that would actually require a bit of energy, all right? So to create what's called adenosine triphosphate, you need extra energy to attach that final phosphate to adenosine. Now you have what's called ATP. And the energy used to create this molecule is still stored there. So in essence, you've created kind of like a battery so that when you need energy later, all you need do is to break this bond, and now the energy can be released and do work. This is important because your body cells require energy. You need energy to make molecules. You need energy to pull chromosomes to opposite sides of a dividing cell. You need energy to make cilia move, and you need energy for muscle proteins to pull against each other when muscles contract. And so this ATP, it's a DNA nucleotide, adenosine, with 3 phosphates attached. Because energy was used to create that last bond, when we break the ATP bond, then that energy is released. And this is the energy that we use to move. It's the energy we use to keep warm. It's the energy which we use to make new molecules, etc. So before we get into the larger nucleic acids, just one nucleotide with some phosphates attached can serve as an energy storage molecule. And your life has required energy. And this ATP monomer, whether we add phosphates to create ATP or we break that last phosphate off to create ADP plus P, this is our way of storing energy and then releasing it when we need it. And so this has essentially been the energy source which has powered your life. So, obviously that's very important. But we don't have to stop there, because that adenosine, that was a small molecule, what we call a nucleotide. And as in some of the other classes, you can take one nucleotide and add it to another one, add it to another one, and to create the long polymer. Each nucleotide has 3 parts: a phosphate group, a pentose sugar, so not a hexose sugar like glucose, but rather a pentose sugar. That's what I'm representing here with those pentagons as opposed to a hexagon. And then a nitrogenous base. So once you have these nucleotides made of these 3 parts, a phosphate, a pentose sugar, and a nitrogenous base, then you can create long chains of these the DNA and the RNA. So here, getting just a little more technical, here you can see our nucleotides. Now, both of these nucleotides have a phosphate, that's one of the parts, a pentose sugar. Notice that there are 5 carbons, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5 in these pentose sugars. And then a nitrogenous base. They're called nitrogenous bases because they have nitrogen in them. That's what those pink atoms are representing, the nitrogen atoms. So each nucleotide is made of a phosphate, a pentose sugar, and a nitrogenous base. And there are 2 types of pentose sugar. Oxygen here. Deoxyribose is identical, but it lacks oxygen there. So you are making a RNA with RNA. If the sugar is deoxyribose, then the deoxyribose and a nucleotide constructing the polymer. Nucleotides, these are small molecules, and by putting them in chains, we then create the polymer. It's a double-stranded molecule. That's what you're looking at here. And each of the this is one in the nucleus of your cells. In your body, you have to go to the moon. This biomodule. I said that essentially proteins are running the cell. All right, now RNA does a lot as well. Is the DNA that we have when we are conceived. So the DNA of a sperm fused with the DNA of an ovum. Now the DNA is organized into different molecules, which we call chromosomes. So here, for example, is the X chromosome, one of the 23 kinds of chromosomes. And there are specific genes located at specific places. And so here is an androgen receptor gene. Remember I said that certain proteins are receptors. So if a cell is to know that, say, testosterone is around, it needs to make a receptor for testosterone. So that's a protein, and here on the DNA is a gene for that. Why? Because you have those 20 amino acids, and you have to know which one comes first, which one comes second, which one comes third. And so that's what the DNA is. The DNA on the left is where there is a code, a chemical code saying when you want to make the androgen receptor protein, this is the amino acid that you'll have to put first, this is the one you'll have to put second. Now, the DNA, which stays in the nucleus, is copied into RNA, which is another polymer, but once again, it has ribose instead of deoxyribose. The RNA goes out to where the protein is made in the cytoplasm, telling the cell which amino acids to put in the right order. If you were to mix this up, if you were to put in arginine instead of leucine, which is the appropriate amino acid, that would be a mutation. And now this protein, which has arginine in it instead of leucine, would not work as it should. And so this could cause a genetic disease. This cell, for example, wouldn't be able to respond to testosterone because its androgen receptor has the inappropriate amino acid because the DNA had made a mistake and informed it to put the wrong amino acid in the sequence. And so if we were to go into multiple cases, as we can later, why would the next patient have cancer? Well, because there is a protein that one needs to make to prevent cancer called P53. But if you're going to make P53, you have to know which amino acid comes first, which comes second, which comes third. That's the information that was present in the DNA when we were conceived. All right, so there are specific chromosomes that have a gene for P53, which has the genetic information telling you what amino acid would come first, what would come second, what would come third. If DNA has a mistake, if one of the nucleotides has been changed, then when the abnormal DNA would then get abnormal RNA. So here's the DNA. The mistake here, one of the nucleotides is produced by a different nucleotide. Nucleotides in the RNA molecule would be correct. So here's the RNA molecule. It would then have cytosine, abnormal there, which would mean that the amino acid order of the protein would be incorrect. The wrong amino acid would be put in place, and now potentially the protein not work, and someone could as a result. So, big picture, this is stuff. I like this stuff. This is chemical stuff. Under the cellular level of organization, we have a chemical level of organization. A cell is just a bunch of molecules, all right, arranged in just the right way. There is nothing here which is simple. It was once thought that molecules could have a mystical force almost, that that you couldn't study biochemistry in a lab because you'd be missing that mystical extra something that makes biomolecules do what they do. We now know that's not true. All right, everything that's here can be described in chemical terms. All right, there's water molecules, there's salts, but then there are also these biomolecules made of carbon. There are carbohydrates, there are lipids, there are proteins, and there are nucleic acids. And as we continue our study of biology and the human form, this keeps coming up. All right, because if you want to study muscles, you're talking about muscle proteins, and obviously maybe the genes which affect the muscle proteins, and the electricity going over the cell membrane of the muscle cell, which is made of lipids, and the sugar that the muscle is using for energy. And so you can't talk about muscle until you have a handle on the biochemistry of it, because then this becomes important. This second video on biochemistry has talked about the proteins which are doing many of the functions of the cell. In essence, they run the cell, but then also going into nucleic acids. And in addition to ATP, which is the energy source of our bodies, nucleic acids include polymers of DNA and RNA, which help to make the proteins with the correct amino acid order, which are then running the cell.